Hello students, welcome to this presentation on rickettsial infections. Genus Rickettsia actually gets its name from this gentleman, Howard Taylor Ricketts, who initially discovered Rocky Mountain spotted fever as a tick transmitted infection. However, this gentleman also lost his life as he contracted the infection during the study. Genus Rickettsiae basically consists of two families. One is family Rickettsiaceae, which comprises of Rickettsia and Orientia, and family Anaplasmataceae, which includes four bacteria, that is Ehrlichia, Wolbachia, Anaplasma and Neo-Rickettsia. For the purpose of further descriptions, I would be focusing more on family Rickettsiaceae as it is endemic in our part of the country. One thing we should remember about genus Rickettsia is that they generally have a gram-negative cell wall, which is composed of peptidoglycan, lipopolysaccharide, and an outer membrane consisting of few proteins. They also have ribosomes for protein synthesis and also possess both DNA and RNA. These features actually differentiate this organism from the viruses, which they were once thought to be so. They are primarily obligate intracellular parasites infecting vascular endothelium and reticuloendothelial cells, and they are transmitted by the arthropod vectors, that is lice, fleas, ticks and mites. Morphologically, all the rickettsial organisms appear pleomorphic and they also appear gram negative. So we say them, generally term it as a pleomorphic gram negative coccobacilli. They are both non-motile and non-capsulated and stain bluish purple with Jamesa and Castaneda stains and deep red with Machiavello and Jimenez stains. They can easily be cultured on the nucleus and the cytoplasm of the infected cells. However, we should note that the spotted fever group of rickettsial organisms grow both in the nucleus and in the cytoplasm when compared to the other rickettsial species. The ambient temperature of 32 to 35 degrees Celsius is generally suited for the growth, and they can be grown in the yolk sac of chick embryo and also in the continuous cell lines, that is both the mouse fibroblast cell line and the HeLa cell line and the Hep2 cell line. Probably their growth in these cell lines might have probably been misinterpreted as viral organisms. So they might have been thought to be viruses, probably because they are growing in these cell lines. Please remember that rickettsial organisms are very highly infectious, and their isolation must be done only in labs having biosafety level 3 facilities. They can also be grown in guinea pigs, mice and arthropods. In fact, there is a reaction which is called as Neil-Mooser's reaction, where we can differentiate certain species of Rickettsia. I will be talking about this reaction a little while from now. this group of organisms are rapidly inactivated by chemical and physical agents. They can be preserved in skim milk, and they are killed by tetracycline, chloramphenicol, and ciprofloxacin. So let us look at the pathogenesis of the rickettsial organism in a nutshell. What you need to be wary of or be remember is that all the rickettsial organisms are transmitted by arthropod vectors. So these arthropod vectors are ticks, mites, louse, and flea. Now, as far as ticks and mites are concerned, they are transmitted by biting and by transovarial transmission. They usually have a reservoir uh, which helps to maintain the organism or the rickettsial organisms in the nature, and they usually pass on to their offsprings. So ticks and mites are primarily transmitted by biting during a blood meal, and louse and flea are transmitted by autoinoculation due to rubbing or scratching of the abraded skin or mucosa contaminated by the insect feces. They are also transmitted by aerosols, that is, by inhalation of the fecal matter, uh, dried louse or dried fleas fecal matter in the lab or as a part of bioterrorism. So that's why I mentioned that it requires biosafety level 3 facilities for lab isolation. Once it bites, it enters the host through the lymphatics, and it spreads from the portal of the entry through the lymphatics, and it multiplies in the regional lymph nodes, and from there it enters the bloodstream. As I mentioned, they primarily affect the vascular endothelial cells, and usually these organisms are intracellular in nature. So they multiply in the nucleus and the cytoplasm. Once these organisms bind to these endothelial cells, they get phagocytosed and remain intracellular. The binding takes place through outer membrane proteins. There are two outer membrane proteins, A and B. After getting phagocytosed and after staying inside the these nucleus and cytoplasm, they multiply by binary fission, taking about 9 to 12 hours. Usually the cell to cell spread occurs by polymerization of the actin filaments, whereas the other actually this actin polymerization takes place only in the spotted fever group, whereas there is another group other than spotted fever group, like the typhus group and the scrub typhus. They accumulate in the cell till the lysis of the cell takes place. Now the reason why they survive inside the cell is because they lack many enzymes that is required for their metabolic purposes, like for the pentose phosphate, purine and pyrimidine pathways. The endothelial cell injury is usually occurring with the lipid peroxidation of host cell membranes, and the damage of the vascular endothelial cells causes the degeneration of these cells and thrombosis of the vessels, leading to rupture and necrosis. Once the bacteria are released from the host cells, they usually die quickly. However, it is important to note that it is in the lymphatic tissues that they survive for longer duration. Now, let us look at some of the rickettsial infections. Primarily, your rickettsial infections are broadly classified as the typhus group, spotted fever group, and the scrub typhus. Now, in the typhus group, you have epidemic typhus, endemic, and endemic typhus. So let us see what is epidemic typhus. Now, epidemic typhus is primarily caused by the organism Rickettsial prowazekii, and it is transmitted by the human body louse, which is called as Pediculus humanus corporus, usually while taking a blood meal from the infected patient. The organisms usually is transmitted by this body louse, and it is strictly adapted to humans. They live in these the louse live in the clothes, and they take blood meal 5 times daily. The lice, as I mentioned, becomes infected while feeding on the blood of rickettemic patients, and Rickettsia prowazekii enters the louse gut epithelial cells and multiplies by binary fission until the massively infected cells burst and are released into the louse feces 5 to 7 days after ingestion. Lice are not adapted to an elevated body temperature and leave the febrile patients for a new host. The rickettsia laden louse feces are deposited on the skin and clothes and are introduced into the new host by being scratched into the louse bitten skin or during the rubbing into the mucous membranes such as conjunctiva. They are also transmitted through inhalation. So, this particular infection is usually noticed in Russia, Eastern Europe and Africa, South America, Asia, and sometimes in India also. However, since 1978, there have been no cases of epidemic typhus reported in Southeast Asia. The incubation period of this particular infection ranges from 5 to 15 days, and generally for all the rickettsial infections you have a classical symptom of headache, myalgia, eye discharge, and rashes. Once the rashes occur after the 4th or 5th day of fever, these rashes usually begin on the upper trunk and then they become generalized. However, classically we should note that the rashes do not affect the face, palms, and soles. So, this is the differentiating feature of rickettsial infections from the other infections that causes fever and rashes. Please remember this particular point. Now, it remains latent in the lymphoid organs, and the myalgia usually is very severe. So, this is the appearance of the louse. Usually this particular infection occurs outbreaks in closed settings like in refugee camps, prisons, and overcrowded communities. Eastern flying squirrels and their lice and fleas maintain this particular organism, Rickettsial prowazekii, in the environment. Now, I told you that this particular organism remains latent in the lymphoid tissues. After several years, there can be a reactivation due to the waning of immunity, leading to sporadic infection or outbreaks. This reactivated infection is called as the recrudescent typhus or Brill-Zinsser's disease. This particular infection can complicate by causing interstitial pneumonitis and mental confusion and coma. The word typhus actually means smoking or hazy, describing the state of mind of the affected patients. Apart from causing the haziness and mental confusion and interstitial pneumonitis, it can also affect the heart, causing myocarditis and acute renal failure. Now, epidemic typhus is usually caused by Rickettsial typhi or Rickettsial felis. This is also seen worldwide, but it can also be seen in India. The reservoir of epidemic typhus is usually rodent, which is your rat, chiefly Rattus rattus and Rattus norvegicus species. There are also other fleas called as Tinocephalus felis. This particular flea is maintained by the reservoirs called as opossums, which are usually seen in Texas and California. The rat flea is also called as Xenopsiella chiopis. Usually the urban ports are rife with rats, and they have been associated with the higher prevalence of this particular infection. The residents and visitors to these urban and suburban regions are at risk when the flea-bearing animals bring the infected fleas into close proximity to humans. The seroprevalence that is demonstrable to the typhus group of antibodies in areas where the disease is endemic, and also in areas where the disease is not well recognized, indicates that this particular infection is usually neglected. The disease is transmitted after inoculation of the infected flea feces into the flea bite wound. Usually the incubation period is about 1 to 2 weeks, and it commonly it is somewhere ranging around 12 days. Because predominantly gut epithelial cells are infected in the flea vector, a reservoir of infected fleas is maintained mostly by horizontal transmission from flea to the vertebrate host to the uninfected flea. Once infected, the flea maintains the rickettsial infection for the duration of its life. That is, it is lifelong. So Rickettsial typhi may also infect the flea reproductive organs, and that is why this explains the low levels of transovarial transmission. The longevity of fleas is unaffected by the gut epithelial cells or disseminated Rickettsial typhi infection. So that's how it enters: bites, feces, inhalation, or through conjunctiva. Compared to the epidemic typhus, the endemic typhus is much more milder, and we can differentiate both the endemic typhus and epidemic typhus by what is called as the Neal-Moore's reaction. Here we inoculate the specimen intraperitoneally into male guinea pigs, and after around 3 to 4 weeks we observe the changes, especially the scrotal inflammation in the animals. So Rickettsii produces scrotal necrosis. Rickettsia prowazekii, or what is called as the epidemic typhus, usually produces only fever without any testicular inflammation, whereas Rickettsia typhi produces the scrotal inflammation, and also it causes adhesions in the tunica vaginalis. So this is how we can differentiate Rickettsii and Rickettsia typhi and Rickettsia prowazaki. So Rickettsia typhi shows a positive Neal-Moses reaction, whereas Rickettsia prowazaki, the reaction is negative. So both epidemic typhus and endemic typhus belong to the typhus fever group. The epidemic typhus, to summarize, is caused by Rickettsia prowazaki and transmitted by louse, whereas the endemic typhus is caused by Rickettsia typhi and is transmitted by flea. Please remember that both of them possess surface protein antigens which are species specific and cross reactive. The outer membrane protein B is usually present in the typhus fever group, and it is coded by the SCA4 gene. SCA stands for surface cell antigen. Now we go to the next group of rickettsial infections, which are called as the spotted fever group. Now there are three infections under spotted fever group. You have Rocky Mountain spotted fever caused by Rickettsia rickettsii, and it is transmitted by ticks. The Indian tick typhus caused by Rickettsia conuri, and rickettsial pox, which is caused by Rickettsia acari and is transmitted by the gamacid mite. Please remember that unlike the typhus fever group, the spotted fever group possesses both the outer membrane protein A and B. The outer membrane protein A is coded by the SCA5 gene, and this particular outer membrane protein A is exclusively present only in the spotted fever group. The protein A helps in binding, and protein B causes cross reaction with surface protein antigens of the typhus fever group. Now we come to the Rocky Mountain spotted fever. Please be informed that this particular infection is a potentially fatal disease. In fact, among the rickettsial infection, this has got the highest mortality rate. This particular infection is usually transmitted by the ticks. There are different species of ticks. In the USA, the species is called as Dermacenter andersoni. In Central and South America, it is Amblyomma caginense, and in Mexico, Arizona and Brazil, you have Rhipicephalus sanguinus. So all are the different species of ticks, and of the three tick stages, the larva, nymph and adult, only the adult dermacenter ticks feed on humans. The prevalence of the pathogenic rickettsia in various tick population, their efficiency of both trans-ovarial transmission and trans-stadial transmission, that means from one stage to the other stage transmission, and their ability to infect the uninfected ticks while co-feeding on the same host are quite variable. The tick transmits the disease to humans during a prolonged period of feeding lasting for up to 1 to 2 weeks. The blood meal and the host temperature associated with tick feeding modulate the expression of certain genes that may upregulate virulence factors of rickettsia. Moreover, salivary products of the ticks enhance the establishment of rickettsial infection. Please understand that the bite is generally painless and it goes unnoticed. Once the tick has attached and it has fed for up to 6 to 10 hours, the rickettsia will be injected from the salivary glands. Sometimes an even longer period may be required for reactivation of the rickettsial virulence in unfit ticks. Humans can also be infected by exposure to the infective tick hemolymph during the removal of ticks from persons or domestic animals, especially when the tick is crushed between the fingers. So, the reservoir host for this particular organism is the rabbits, dogs, and the rodents. And like in the other rickettsial infections, you have headache, fever, rash, abdominal, and joint pain. The rashes are typically characteristic and they appear on the extremities, that is, the wrist and ankles, and in the trunk. The complications usually include vascular damage, increased permeability, edema, hemorrhage, DIC, that is disseminated intravascular coagulation, along with the involvement of the CNS and the lungs in the form of interstitial pneumonitis and myocarditis and renal failure. So this is the typical distribution of the rash of Rocky Mountain spotted fever. Now the Indian tick typhus variety is caused by Rickettsia conuri, and it is transmitted by the tick species Rhipicephalus sanguinus. Okay, so clinically, in this particular variety, the Indian tick typhus variety, you will observe an eschar. This eschar is observed at the site of the bite of the tick, and the disease is more severe in patients with comorbidities such as diabetes, alcoholism, or heart failure. So, basically, it is also present in other areas apart from India. So, depending on the geographical locations, you will have different names for these illnesses. So, you have African tick bite fever, that is caused by Rickettsia africae. You have Japanese spotted fever by Rickettsia japonica, Queensland tick typhus, which is seen in Australia due to Rickettsia australis, and different species of ticks are present for each of these different infections. The reservoir is usually the rodents, and as I already mentioned, the mode of entry is through the bites and also by the crushing of the the feces. Now, rickettsial pox is a third group of the spotted fever group, and rickettsial pox is caused by Rickettsia acari. Usually, it is seen more in USA and Russia. The vector of Rickettsia acari is the gamacid mite. It is very important to remember the difference between the ticks and the mites. The ticks are usually seen with the naked eye, and they usually expand up to 3 centimeters in length after feeding. But comparatively, mites are more microscopic and they are very difficult to see with the naked eye. All of the ticks are parasitic, but mites, some of them are free living and some of them are parasitic. Usually ticks lay its egg on the ground in cracks or under the rocks, whereas mites usually lays its egg in tunnels at the site of infection on the body of the host. So, rickettsial pox is transmitted by the mite species Leponisoidus sanguinus. The reservoir is mites mice, which is mus musculus. You have to remember here that rickettsial pox also causes eschar, like the Indian tick typhus variety. The eschar is nothing but a painless black crusted lesion surrounded by an erythema, which is present at the site of the bite. So, the classical clinical manifestations of rickettsial pox are that it causes vesicular rashes or varicelliform rashes, almost resembling to that of chicken pox, along with regional lymphadenopathy. So, the features that distinguishes this particular infection from the other rickettsial infection is that you have vesicular rashes, eschar, and regional lymphadenopathy. This particular infection is also endemic in USA, Ukraine, Turkey, and Mexico. So, this is the characteristic rash. Earlier, this particular genus was Orientia was called as Rickettsial susugamushi, but currently, due to the cell wall composition and due to the genetic variation, this particular organism, Rickettsial susugamushi, has been renamed as Orientia susugamushi, and this particular organism causes the infection scrub typhus. The scrub typhus is usually affecting the military population and is usually associated with war, because the organism is transmitted by the trombiculoid mite, which is usually found in these areas where there are lot of scrub vegetations, consist of low-lying trees and bushes, and also in the sandy and semi-arid mountain desert areas. So the trombiculoid mite is the vector and the reservoir is the rodent or the bird. There are 5 major antigenic types identified with scrub typhus. They are called as Boreon, Gilliam, Karp, Kato, and Kawasaki. And because of this antigenic diversity, the immunity usually decreases after 1 to 3 years. Epidemiologically, there is one term called a zoonotic tetrad, which is quite commonly associated with scrub typhus. This we have to remember. There are 4 essential elements that are required to maintain Orientia susugamushi in nature. One is the trombiculoid mite. Second is are the small mammals like the field mice, rats, which are the reservoir mammals. Then you need the scrub vegetations in the forest, and also the wet season where the mite lay eggs. A particular geographical area called a Susugamushi triangle has been described where this organism or this infection is prevalent, and this triangle extends from northern Japan and far eastern Russia to northern Australia in the south and Pakistan in the west. So countries such as Japan, China, Philippines, India, Pakistan, Afghanistan, tropical Australia, New Guinea, and Pacific Islands are endemic to this particular infection. In the Indian context, this particular infection has been isolated right from the sub-Himalayan belt of the Jammu to Nagaland, Himachal Pradesh, Sikkim, Darjeeling, Bihar, Rajasthan, Maharashtra, and in southern India from Pondicherry or Puducherry, Karnataka, Tamil Nadu, and Kerala, especially during the cooler months of the year. Though it is mainly reported from rural areas, recently there have been cases of scrub typhus even from the urban areas. Among all the stages of the mite, of the trombiculoid mite, the larval form, that is the chiggers, are the only stage that feed on the humans. The genus Leptobromedium or is this mite species that usually infects the humans. So there are two particular variants. You have Leptobromedium akamushi, which is seen in Japan, and the species of mite seen in India is Leptobromedium daliensis. So that is the difference in the species of the mites, and usually it is maintained, the transmission is maintained among the mites by transovarial transmission. So the incubation period of this particular illness is around 1 to 3 weeks, and it is usually found in the scrub vegetations. So therefore you have classical manifestation which is called as eschar. Please remember, among the rickettsial infections, we have noted so far that eschar is seen typically in the Indian tick typhus variety, in rickettsial pox, and this is the third in rickettsial infection where eschar is usually seen. So the classical triad of clinical manifestation is the eschar at the site of the bite, regional lymphadenopathy, and maculopapular rash. It is seen only in 40 to 50% of the cases. So it is very important to note these eschars, because the eschar may be present in any of the anywhere in the body. So whenever we infect check, or whenever we take a clinical history from patients with fever, with rashes, etc., we should always look for the evidence of the bite of these vectors in the form of eschar. So that is very, very important as far as scrub typhus is concerned. In severe cases, you have encephalitis and pneumonia. Usually lab diagnosis is done by animal inoculation into the male guinea pigs and mice. As I already mentioned, we have the nail mousers reaction, which can differentiate between Rickettsia prowazaki and Rickettsia typhi. Please also be informed that along with Rickettsia typhi, a positive nail mousers reaction will also be given by other species like Rickettsia conori and Rickettsia akari. Egg inoculation and cell culture method in Vero cell line and MRC5 lines are also other cell lines which are used to isolate this organism. Now we come to one of the very important non-specific test, which is called as the Weil-Felix test, because usually serology is the mainstay of diagnosis of rickettsial infections. So you have non-specific serological test and specific serological test. Rickettsia, the non-specific test for rickettsial infection is called as the Weil-Felix test. As you all know, this is a heterophil agglutination test. That means you are using the antigen belonging to some other organism which shares the epitope of this particular target organism. So in Weil-Felix test, we generally use the Proteus strains, that is OX19, OX2 and OXK strains of Proteus, because they share a common group specific alkali stable lipopolysaccharide along with the rickettsial organisms. So here rickettsial antibodies are detected by using the Proteus antigens, and it is generally a tube agglutination test where serial dilutions of patient's serum are made, and it is treated with the non-motile strains of Proteus vulgaris, that is OX19, OX2 and OXK. the reason why it is called as the non-specific test is because of the results which you are seeing here. Now, in Rickettsial typhus, okay, in the epidemic typhus and endemic typhus, your agglutination is mostly seen with the OX19 sera. Okay, so both the epidemic and the endemic typhus, the agglutination is seen more with OX19, whereas in the tick-borne or the spotted fever group, you will see both OX19 and OX2 sera getting the antibodies which are present to both OX19 and OX2 sera. Scrub typhus is classical, where you will see the antibodies to OXK. The test is usually negative in rickettsial pox and ehrlichiosis. And please remember that the sample should be collected only after 5 to 7 days of the onset of fever. And you can do both a quantitative and qualitative test, where a titer of 1:80 is considered to be possible infection. False positive reactions are seen with Proteus infection, but if you are able to demonstrate a fourfold rise in antibody titer in paired sera, it becomes more suggestive of a rickettsial infection. False negative reaction is seen with prozone phenomenon, which is the phenomenon which is associated with any agglutination test. So here the lattice-like network is not formed because of the excess of antibodies in the patient sera. So if you do not dilute the patient's serum properly, the test will appear negative even though the antibodies are present. So therefore, the serial dilutions of patient sera should be always carried out. Once the Weil-Felix test gives a positive reaction, then we go for specific test, which is the indirect immunofluorescence assay. The antibodies here appear only after 7 to 10 days of infection. Therefore, a titer of greater than 1:64 is considered significant. We can also do an ELISA testing, that is, we can do IgM capture ELISA, which has got a very good sensitivity and specificity. So other methods, as I already mentioned, are by isolation in the cell lines and also by the Neal-Moses test. A word about molecular reactions. PCR, the real-time PCR format are available for detecting the specific rickettsial DNA to certain genes which encode the surface antigens, and also the OMP genes or the 16S rRNA genes or the heat shock protein GROEL gene. The specimens useful for molecular testing is whole blood, buffy coat, skin rash biopsy, lymph node biopsy or tissue specimen. Again, the PCR is usually positive within the first week for blood samples, because rickettsial organisms are detected usually in the first 7 to 10 days. So how do we treat this infection? The treatment is generally carried out with doxycycline given as 100 mg twice a day orally for 1 to 5 days. For scrub typhus, let me also remind you that serology is the mainstay, and IgM antibodies appears by first week and IgG appears by second week. Whereas if there is a reinfection for scrub typhus, the IgG can be detected earlier, as early as the 6th day of infection. So how do we prevent these infections? These infections are generally prevented by control of the vectors. Now, as far as ticks are concerned, it is better to avoid contact with the ticks by using repellents and protective clothing. Especially permethrin treated clothing is effective at reducing the number of tick bites. Um, and another very important factor that we need to note here is a personal protection. Now, proper clothing should be used to limit the access of ticks to the skin and thereby prevent bites. Pants should be tucked into the boots or socks, and the shirt should be tucked into the pants. We should avoid sitting or lying on scrubby areas. Avoid hanging clothes on scrubs or trees. We have to disinfest the pets regularly. Long sleeves will help, and a hat will be useful if you are crouching or crawling in bushes or undergrowth. Light colored clothing should be used to make the ticks much easier to detect. Now, repellents that are used, you can use repellents like permethrin, as I mentioned already, and also DEET, DEET, that is NN dimethyl M toluamide or NN dimethyl 3 methyl benzylamide. So, DEET 33% repellents can be used, and these repellents provide protection for up to 12 hours depending on the environmental conditions. It should be applied in a thin film over the exposed skin surfaces, and permethrin should be applied to the lower pant legs, crotch, waistband, shirt sleeves, collar, uh, from placket and lower edge of shirt, and it should never be applied directly to the skin. Permethrin aerosol contains 0.5% permethrin and should be liberally sprayed to the point of dampness over the entire outside surface of the uniform. So, where acceptable, uh, we can, environmental management is also very important here. So we have to remove low growing vegetation and brush or scrub areas. So this eliminates the structural support that ticks need to contact the host and thereby reduce the incidence of tick attachment. Now we can remove the leaf litter and underbrush, and also this will also reduce the density of the rodents, the deer, mice, etc. And moving lawns and other grassy areas to less than 6 inches reduces the potential for the tick contact. So when environmentally acceptable, we can do controlled burning. And another habitat modification technique is to thin early successional shrubs and grasses, and stressing the overwintering tick population and reducing the survivability. Chemically, we have to ensure the clearing of vegetation by spraying malathion spray, especially when outdoor camping of troops. And also this malathion spray is known to be a chigger bite repellent and can be used to before venturing into the forest areas for any of these activities. So I hope you have understood the rickettsial infections in general. So just to summarize the rickettsial infections, so far we have the typhus fever group, spotted fever group, and scrub typhus. The typhus fever group, you have epidemic typhus transmitted by louse, the endemic typhus which is transmitted by flea. You can differentiate both these groups by the characteristic appearance of the rashes. The rashes are seen more in the trunk in the endemic typhus, and in the epidemic typhus you see all over the body, and characteristically all the rashes in rickettsial infection spares the palms and soles. The spotted fever group is caused by Rickettsia conori, Rickettsia africae, and Rickettsia acari. So Rickettsia conori causes the Indian tick typhus, Rickettsia acari, rickettsial pox, and one more species you have Rickettsia rickettsii, which causes Rocky Mountain spotted fever. So out of all these infections, RMSF is the most causes severe mortality among all the species. And then you have scrub typhus group caused by Orientia susugamushi. Now, please remember that Susugamushi is actually Japanese name. Susuga means dangerous, Amushi means insect. Okay, so that I actually forgot to mention. Um, scrub typhus is caused by Oriental Susugamushi, and it is transmitted by trombiculoid mite, whereas your rickettsial pox is transmitted by gamacid mite. The trombiculoid mite belongs to the Leptobromedium species. And as far as the diagnosis is concerned, we have non-specific test, that is the Weil Felix test, where OX19 is positive for the typhus fever and the both your endemic and epidemic typhus. In the spotted fever group, OX19 and OX2 are positive, and the scrub typhus group, OXK is positive. So the specific diagnostic test include indirect immunofluorescence test, ELISA test, and PCR. Thank you for your patient hearing.